How does an organism grow? How do cells divide? The answer to both of these questions is mitosis. Mitosis is the process by which cells divide. Genetics uses an approach called cytogenetics to study the formation and structure of chromosomes when cells are in different stages of mitosis under a microscope.
In a typical cytogenetics lab, geneticists must first prepare samples of cells from their source of interest, such as a patient's tissue, blood, and bone marrow, before they can analyze the chromosomes within the cells of their sample. Today, we are going to be modeling mitosis using a very special kind of cell, an Oreo cookie.
Although we're talking about cell division, cells actually don't divide all the time. Most cells in a given sample will actually be in a phase called interphase. In this phase, cells can carry out their normal cellular functions and prepare to divide. There are 3 stages of interphase: G1, S, and G2. In G1, cells grow physically, replicate their organelles, and synthesize proteins. In the S phase, the cells will replicate their DNA. Lastly, in the G2 phase, cells will continue to grow and organize their cellular contents. Throughout interphase, the individual chromosomes cannot be seen since the DNA is still coiled around histones as chromatin, and it is not compacted into its chromosome form, which is usually depicted as an X.
The first stage of mitosis is prophase. Here, the chromatin condenses into chromosomes that are distinguishable under a microscope. Each chromosome can be seen as 2 sister chromatids, and the centrioles, which are the microtubule organizing center, begin to move to opposite ends of the cell.
During metaphase, microtubule spindles will protrude from the centrioles and attach to the centromere of the chromosomes. At this point, all of the chromosomes will be lined up at the center of the cell.
Next up is anaphase. During anaphase, the spindle fibers attached to the centromeres will contract, pulling the sister chromatids apart and moving the daughter chromosomes to opposite ends of the cell.
Once the sister chromatids have reached the opposite poles of the cell, they will start to unwind and become longer and thin again. The spindle fibers that helped pull the chromosomes apart in anaphase will start to disappear. At the same time, a new nuclear membrane will form around each group of daughter chromosomes.
Finally, the cell reaches cytokinesis. During this process, the cell fully cleaves into 2. In animal cells, this is accomplished by a contractile ring that constricts the cytoplasm. In plant cells, a new cell membrane forms right in the middle of the cell before the cell secretes material to build 2 new cell walls.
But what happens if mitosis goes wrong and the cells get stuck in one stage?
When a cell is exposed to certain drugs or chemicals, it can cause disruption of the cell cycle. For example, colchicine, which is an anti-mitotic drug, binds to the tubulin and prevents the elongation of the microtubule polymer. The disruption in microtubule polymerization results in the inability of sister chromatids to segregate during mitosis. If one were to quantify the mitotic index, which is the number of cells within a sample that are in mitosis, based on the chromosome structure that we observe underneath the microscope, we would expect a sample treated with colchicine will possess more cells that are stuck in mitosis, and thus a higher mitotic index compared to a control sample that is not treated with colchicine. The cells are therefore arrested or stuck in metaphase. In real life, such antimitotic drugs are used in some cancer therapies as they prevent tumor cells from dividing and ultimately lead to cell death by apoptosis.
We hope you enjoyed learning about the many stages of mitosis and how the cell prepares and undergoes cellular division. All eukaryotes owe their growth to mitosis, so it's really important to know what happens when mitosis goes wrong.